Okay, sorry everyone, I'm going to go ahead and get started. As many of you are aware, functionally related and phenotypically similar traits often evolve in tandem. And this correlated evolution can be due to pleiotropy, genetic linkage, or correlated selection. And these genetic correlations are important because genetic correlations can constrain adaptive evolution if responses to selection on one trait cause maladaptive trait changes in correlated traits. So the traits that I'm going to be considering today and thinking about in the context of correlated evolution are pathogen resistance at juvenile and adult host life stages. And I'm wondering whether basically resistance at these two stages function as separate traits. And considering the evolution of age-specific resistance is important because juveniles are often more susceptible to pathogens than adults. And this has important epidemiological consequences because juvenile susceptibility often drives epidemics in natural populations across taxa. So my overarching study question is, are juvenile and adult resistance separate traits that can evolve independently? And to assess this question, I'm using the study system anther-smut disease and its host, Silene latifolia. It's plant host Silene latifolia. So anther-smut disease is a fungal pathogen in the genus Microbotrium, and anther-smut sterilizes its host plant and induces the host plant to produce infectious spores on the anthers instead of pollen. And then it can be transmitted by insect pollinators to healthy plants. And then it induces a systemic infection in the plant host. And if the plant host is a female, it can sterilize and masculinize the female. And then because this is a fully sterilizing disease, and because plant hosts rarely, if ever, recover, anther-smut is a highly virulent pathogen that imposes strong selection for resistance. Okay, so there are two transmission modes for anther-smut. There's, as I said, there's a sexual transmission mode in which pollinators can carry spores from healthy flowers, from infected flowers to healthy flowers, but there's also an aerial transmission mode in which infected plants can drop spores on nearby seedlings. And modeling research in Emmy Bruns' lab, the lab I did my postdoc in, suggests that up to 80% of total transmission events are to seedlings. Okay, so I predicted that if juvenile and adult resistance can evolve independently, then they should be genetically uncorrelated. Furthermore, I predicted that selection on resistance at one life stage should not affect the evolution of resistance at the alternate life stage. So to test my first prediction that juvenile and adult resistance are genetically uncorrelated, I measured the resistance of 45 different full-sibling Silene latifolia host families. So you can think of the 45 host families as different genotypes. And I measured their resistance at 4 different ages, 9 days, 19 days, 78 days, and 104 days. And then to measure resistance, I inoculated each plant. In the case of when I was inoculating the seedlings, I did a small pipette inoculation. And then when I was inoculating adults, I sprayed them with inoculum until dripping. And then I reared the plants until they flowered, scored the plants phenotypically to determine which plants were infected and which plants were uninfected. And then I calculated the proportion of plants that remained healthy following inoculation in each of my genotypes and used that as my metric of resistance for each genotype. Okay, so here on these plots, I'm going to show you my measure of resistance, and in each plot, the younger age is on the x-axis, and then the older age is on the y-axis. Each point on the plot is a single host family. And you can see that this first plot compares 9 days versus 19-day resistance. So you can see that my two juvenile ages measures of resistance were highly positively correlated. And similarly, my two adult age 78-day resistance was positively correlated with 104-day resistance. So adult resistance was correlated with adult resistance. And then, however, if you look at all possible pairwise combinations of juvenile resistance measures versus adult resistance measures, you see that there was no correlation. And so this suggests that juvenile resistance is uncorrelated with adult resistance. To test my second prediction that juvenile and adult resistance can evolve independently, I used experimental evolution. So in my experimental evolution regime, in one treatment, I exposed host plants as juveniles, reared them up until they were reproductive, and then I crossed those plants that didn't get infected to select for juvenile resistance. In a second treatment, I exposed plants as adults and then waited for the infections to take, crossed those plants that didn't get infected to select for adult resistance. And then in a third treatment, plants were mock inoculated and randomly crossed, and that third treatment functioned as a control. Okay, then following this experimental evolution regime, I assayed all of these evolved populations for their juvenile and adult resistance. I predicted that if juvenile and adult resistance are regulated by the same mechanisms and function as the same traits, then selection for juvenile resistance should lead to an increase in juvenile resistance and also an increase in adult resistance. And similarly, selection for adult resistance should lead to an evolved increase in adult resistance, but also an increase in juvenile resistance. On the other hand, if juvenile resistance and adult resistance are regulated independently, then selection for juvenile resistance should lead to increases in juvenile resistance with no change in adult resistance, and selection for adult resistance should lead to increased adult resistance with no change in juvenile resistance. Okay, and then I also wanted to mention that because of that, I was able to add another treatment to all of these passaging regimes. I was able to take the plants that became infected, temporarily heat cure them by treating them with high temperatures in a growth chamber, and then that clears the infection and allows me to cross them, so I could cross the infected plants to select for susceptibility at each age. Okay, so here I'm going to show you the experimental evolution results from the juvenile assay. So here I have the proportion healthy following juvenile inoculation on the y-axis. So this is, again, my metric of juvenile resistance. And then across the x-axis, I have each of my experimentally evolved population treatments with the controls on the left. And then the ancestor is this dashed line across the bottom. And what you can see, and then each point is an independently evolved replicate population. And what you can see is that there was no significant effect of experimental evolution treatment on juvenile resistance, although there was a trend toward higher juvenile resistance in the experimental populations that evolved under selection for juvenile resistance. That's this bar. Okay, on the other hand, now when I look at the adult resistance of the evolved population, here I have proportion healthy following adult inoculation on the y-axis, so that's my metric of adult resistance. And then the controls on the left, and here you see that adult resistance, the populations that were under selection for adult resistance evolved significantly higher adult resistance as expected relative to the controls. On the other hand, populations that were selected for juvenile resistance did not significantly differ from the control. So this is what you'd expect to see if juvenile and adult resistance can evolve independently. Okay, so overall, in conclusion, I've shown that juvenile and adult resistance to an infectious pathogen are uncorrelated across host genotypes. I've shown that pathogen resistance at different host life stages can respond to selection independently. And then taken together, these results suggest that juvenile and adult pathogen resistance likely function as independent traits that can be regulated by different mechanisms. Okay, in the future, I'd like to sequence my evolved populations to evaluate how the genome evolved in response to selection on juvenile versus adult resistance, and also to do transcriptomics to evaluate how does the transcriptome respond to exposure to the pathogen at the juvenile versus the adult age. And then I think taken together, with these transcriptomics and genomics projects could allow me to identify what loci are important for conferring juvenile resistance versus conferring adult resistance, and is there a lot of overlap between those loci. And I'd also like to use these evolved populations in experimental field transmission studies to evaluate basically how does juvenile versus adult resistance affect pathogen spread in experimental populations. Okay, with that, I'd like to thank everyone who contributed to this work, especially my postdoc advisor and collaborator, Emmy Bruns. I'd also like to make a quick plug that I'll be recruiting master's students to my new lab at the University of New England, and I'd be happy to take any questions. Thank you for your time. Mandy.
Do you have any thoughts on why that response to selection in the juveniles was, well, maybe not less, but so much more variable than the response in the adults?
Yeah, that's a good question. I think, first of all, I'd say, I don't believe that juvenile resistance can't respond to selection. I think we're probably dealing with a power issue. So, this is a hard system for experimental evolution, so we only had 3 replicate populations and we only had 3 passages. So I'm not trying to make the conclusion that juvenile resistance can't evolve. Now, why did it evolve so much less than adult resistance? That I don't know. I didn't expect to see that. We do have lots of variation across genotypes in juvenile resistance. So I expect that juvenile resistance should be able to evolve quickly in response to selection, and I'd love to know why we saw a stronger response to why we saw more response to in adult resistance than response in juvenile resistance.
Cool. So, hey everyone, good morning. So my name is Luis, and I'm going to present you a bit of some of the projects I've been working while in my postdoc in Neuchâtel. Now I'm in British Columbia. So this is past work, but it still connects with what I'm going to work. And what I'm going to talk a bit is the relationship between parasite transmission and virulence evolution. That is highly debated topic in evolutionary biology. So as most of you know, hosts or pretty much any life form is parasited in some kind of way. A parasite exerts some kind of cost into the host. We usually talk about virulence as a cost of infection or cost of the parasite or the immune response altogether, and as a proxy often we use infection mortality, for example. And this is a very important property of infection itself, and it has been highly important for medicine, conservation, and so on, because it helps us predict how diseases are going to evolve. So how virulence is supposed to evolve, it's based on this quite old at this point theory called trade-off theory of virulence evolution, that basically it's around 40 to 50 years old, and it basically is the foundation of all WHO guidelines, medical, etc. And what the theory it says, it's basically that with an increasing virulence, you should have a trade-off with parasite transmission, that here in this graph is called parasite fitness. But parasite fitness is basically transmission, because reproduction is basically one of the most important fitness measures. However, in the last 10, 20 years, we have seen growing objections against this theory, and I'm going to name some of them, at least the more pertinent ones. for example, does not represent most parasite transmission cycles. For example, doesn't include vector transmitted diseases, doesn't include obligatory killer parasites, etc. And there's very few, actually, wild and lab evidence to support this theory. In part because it's very difficult to find parasites that will have a high virulence and no transmission, because that's going to be counter-selected. So when we looked at this, we thought, okay, how about we disrupt what transmission and we try to select for example, 2 different times of transmission within a host. So for that, we took advantage of our lab host. That is Anopheles gambiae, so it's the malaria vector. And for those that don't know the life cycle, basically it has terrestrial step phase, and then it has 3 phases, there are 3 stages that are aquatic. So the adult basically leaves the eggs in the water, the eggs will eclode into larvae, the larvae will develop in the water where they will eat bacteria, plankton, and then they will develop into a pupa that then it will emerge into an adult. And then the adult just flies away and etc. And the parasite that we decided to test this hypothesis and evolve was Vibrio culicis that was previously presented by Tiago. It's a microsporidian and it has quite interest because of to repress its malaria. So how it works in the life cycle, its transmission cycle coincides with this mosquito life cycle. So basically, first, the spores from Vibrio are ingested in the larval stage. As you can see, I don't know, do I have a pointer? Yeah. They are ingested by the larvae. They basically fixate into the gut cells, and they produce basically spore factories, spores that are going to be infective, but they are not released that soon usually, because they have to be produced in a mass scale first. And for that usually happens in the adult stage, where the spores start getting released into the body of the mosquito, and whenever it dies, then it will spread the spores basically. And usually has to be in an aquatic or in a water body, because these spores are very sensitive to desiccation. So that's the transmission cycle of the parasite. And so after this, we thought, so how does time to transmission actually affect virulence evolution? Are they linked? And does this go with the theory of virulence evolution? So for that, we created 2 different regimes, early, what we call early and late. And basically what we have here, it's a survival curve of the death by the microsporidian parasite that I was evolving. And what we can see is that we decided to select the first third of the mosquitoes that died and remove their parasite, and the last third of mosquitoes that died and removed their parasites. So the early one is basically selecting for fast killing or a shorter within host time. And the last one is selecting for a slow killing and a longer time within the host. So a priori, we would expect that actually early would select for some kind of more virulent parasite, and late would select for something more like within the lines of mutualism, if we see it as a gradient between mutualism and parasitism. Interestingly, when we look at the parasite response to selection, we actually saw the opposite. So we saw that late the parasites that killed last were actually much more virulent in a common garden experiment. And we were a bit surprised by this, but we were even more surprised by the fact that they also developed much faster than any other regime. So in here I added also the reference stock, so this is the ancestral of the parasite. And as you can see, this is basically in the y-axis, you have the proportion of females that already are secreting infective spores. And you can see that for the reference stock, for the control, you take 10 days for all the females to are releasing spores. But the same cannot be said about the late one, where at day 3, all the females are already releasing spores. More interestingly, the amount of spores that they are releasing is also much higher for the more virulent. The more virulent you are, the more spores you produce. So the more you actually exploit the host. This exploitation, we recently found out that is in part due to a better sequestration of the iron that is circulating within the mosquitoes, but I won't delve into that in this presentation, but if you are curious, I could go into more detail about that. And after seeing this, we thought about, okay, if this parasite is only transmitted upon death, the load that the mosquito has, in a way, it's infectiousness. So if you plot infectiousness against virulence, what we have is this beautiful kind of straight line that is basically a positive relationship between the parasite load or infectiousness with virulence. That is actually totally the opposite of what is predicted by the theory and has been seen across dozens of publications. So why these unintuitive results? So we went back to the trade-off theory and we started looking a bit into literature, and we found that there's a huge ambiguity in what is a parasite transmission, because obviously different people work on different steps of infection process, and transmission can be transmission in the new host, transmission in the first host, or could be just survival between hosts. So all of this is a bit confusing, and so that led me, Jacob, and Kyla we wrote this opinion piece where we tried to combine all of these into one framework and have a proper nomenclature that everyone can use, and there's no confusion about what actually transmission is at different steps. Because so far I've been talking about within host evolution, and that's one step of transmission, but it's not all of the picture. The whole picture actually includes within host evolution, but also among hosts. And besides STDs, no other pathogen actually does not have some kind of interhost environment. All pathogens have an interhost environment. And then there's also host 2, of course, that has its own set of different immune responses. It can be tolerant, resistant, susceptible, so I won't delve into that because that's another talk for another day, but I will delve a bit more into the interhost environment because I think it's particularly important and heavily neglected, and it got a bit of attention with COVID, with the droplets, etc. So we took a focus on the interhost environment, and in this opinion piece, we kind of thought about the different factors that can affect the survival or the transmission potential of a parasite during this interhost environment, and we separated into two. What we call QP, that is basically intrinsic factors for the parasite. For example, parasite quality. So for example, if the mother cell produces a lot of spores, the quality of the spores is probably low because you cannot separate as much resources. You have to separate much resources for more cells. You have less time to produce them. Errors happen, etc. And we also have extrinsic factors like humidity, temperature, like I just mentioned. This parasite is particularly sensitive to UV and also to low humidity. So that will also affect its survival in the interhost environment, and therefore it will affect its transmission into the new host. So we took this in consideration and we asked, so how does this evolved parasite that we just evolved perform in the outside host environment? And for this, and also how does it relate with the virulence? Because we were hoping that it somehow fits with our original theory and help us understand better the results we got. And for this, we ran an experiment where we had a host that produced spores. So all the spores had a given age. And then they were stored at 4 degrees. That is basically the temperature they are stored at the lab. So they are adapted for that for at least 10 to 20 years, and then 20 degrees. And we kept them for a long time period, and then at 0, 45, and 90 days, we just infected new naive populations of mosquitoes to see if they were able to infect them, to have like a proxy for mortality of these parasites. And interestingly, what we got is that the parasite's performance, and here we have in the y-axis the same kind of response, so proportion of females with spores, so females that were infected and they were secreting spores, and then we have in the x-axis we have time, and what you can see is the dashed lines, so the ones that were 20 degrees, they all have lower infectivity compared to the solid lines, so temperature has an effect irrespective of parasite treatment. But more importantly, when we look at the parasite treatment, so early, late, or the reference stock, what we saw is that actually the ones that are very virulent, so the late, for example, the purple one, you can see that is the one that falls more drastically with time. So although they are super virulent within the host, they really do not survive outside the environment. So they have what we call lower spore quality. And why do we know that is lower spore quality? Because we measured it by these concepts that I mentioned before, QP and QE, and we have here basically a correlation between parasite quality and the virulence of each of the different parasites, and we have a negative correlation, meaning that basically the most virulent ones have lower parasite quality. So, very quick concluding remark. Okay, so I should finish. Okay.
Hi, good afternoon. My name is José Salomé-Correa, and I'm a PhD student from NYU. And today I will be presenting my research topic on the construction of recombinant inbred lines as an evolve under sequence experiment to study inbreeding, dominance, and recessive deleterious variation. So, this is moving forward. Okay, there we go. Okay. So dominance refers to the relationship between a dominant allele and a recessive allele, where dominant alleles mask the deleterious effects of recessive alleles. So during outcrossing, these rare recessive variants will tend to accumulate throughout the genome as heterozygosity masks their deleterious effects. During inbreeding, these deleterious variants become homozygous, and the deleterious effects become exposed. If these deleterious effects are strong, they will be purged from the population. And if they're moderately deleterious, they will accumulate and decrease the overall fitness. So dominance is difficult to study because these relevant variants are difficult to access, as outcrossing populations have more rare recessive variants but have high genetic load and inbreeding depression, whereas inbred populations have fewer deleterious variants due to purging. Another reason is that statistics is difficult because of their inherent features. Given that they are low frequency, they have small effects, they're conditional interactions due to the relationship between dominant and recessive, and they contribute too little additive variance to be detected. Therefore, what we want is an outcrosser with a large number of rare variants but low inbreeding depression, and also a system that allows us to control frequency and homozygosity of rare variants. So what we want is to be able to predict the degree of fitness reduction due to homozygosity. And we want to have a data set in which we can have a varying range of genome-wide homozygosity, and also fitness measurements, which we can then use to observe and predict the phenotypic effects that occur whenever you homozygose certain alleles. So our approach is to do crossings between inbred lines of varying degrees of relatedness. And here on the far left, we have different recombinant inbred lines that have varying degrees of relatedness, shown here by having the same colored haplotype blocks. And some of them have the same haplotype blocks at the same positions. So what we can do is that we can grab those crosses and select for homozygous states for those particular locations. So what we are doing is to do the replicable crossing of inbred lines to study variants in homozygous and heterozygous states in many different genetic backgrounds and fitness-related traits. So what we want is to have an outcrosser with lots of variants, but we're also wanting something that we're able to inbreed to create recombinant inbred lines. And our model here is Cinerabditis bci, which is a wild outcrossing with good biological resources. This nematode is found in an island in Panama called Barro Colorado Island, and it is outcrossing, and we're also able to collect lines and cryopreserve them. And given that it's a Cinerabditis, all of the resources that were developed for C. elegans can be used with them. And another reason is that we have a high-quality chromosome scale genome assembly with annotations. And here's what the genome of CBCI looks like. It's pretty small. It's around 93.7 megavispers, and it has a lot of variation, around 1.5 million SNPs. And it has high synteny with C. elegans. And the main reason is that CBCI has low inbreeding depression, allowing for the construction of recombinant inbred lines. In here, we have a SIP mating assay where on the x-axis, we have the generations of SIP mating, and on the y-axis, we have the fractions of surviving lines, showing the lines that are surviving for different generations of SIP mating for 3 different species of outcrossings in Cinerabditis. In here, we observe a large number of line extinction in all of these species. However, with Cinerabditis bci, we have around a third of those lines being able to survive, which allows us to have a manageable level of inbreeding depression that allows us to create healthy inbred lines. So our topics for today is for me to convince you to establish that our real panel is a good resource for quantitative genetics to study dominance, and I'm going to go over the characterization of real haplotypes, as well as the description of real fitness-related traits. So first I'm going to go over the construction of recombinant inbred lines. Our panel draws its genetic variation from the wild. We created 3 founders from 6 wild worms, and our founders segregate 6 haploid genomes. And then with those founders, we created a starting population by making crosses between 1 male founder with 2 unrelated female founders to create a population of millions of individuals. And each of these populations segregate the 4 founder haplotypes. Then from this starting population, what we did is that we graft pairs of individuals and performed SIP mating for 25 generations. And we were able to create over 420 recombinant inbred lines and sequenced all of them. And then using a real construction approach, we are able to control homozygosity and frequency of those rare variants by the controlled crossing, given that each reel has a unique set of haplotypes blocks, and both panels are half-siblings, and fitness of reels and reel crosses are replicable. Therefore, we can use our reel panel to control inbreeding through crosses of reels with varying relatedness, make low-frequency variants more common, control homozygosity, and increase statistical power for mapping. So the replicable crossing of reels allows us to study variants in homozygous and heterozygous states with many different genetic backgrounds using quantitative genetic methods. So what we want to know is what are the 6 wild haplotypes that are being segregated onto our founders. So we have genotype data for the entire panel for pools as well as for every single recombinant inbred line. And we use the founders, we use the G5 starting population to genotype our founders, and we use our reels to phase our founders. It's kind of a complicated method, and I don't have the time to explain it, but if you guys want to go over it, I'd be happy to talk to you later. Knowing that, assuming that we know that, how are these 6 founder haplotypes segregating into our reels? And this is what they look like. On the left, we have chromosome 2 and the haplotype blocks for our different recombinant inbred lines. Each horizontal line represents a recombinant inbred lines from these 2 different crosses. And the different colored segments represent the founder haplotype blocks that go into them. So in here, we observe that there is lots of haplotype blocks on the ends of the chromosomes, and the middle of the chromosome has fewer. And there are around 5 recombination events on our chromosomes for the creation of recombinant timbrate lines. And these patterns mirror the patterns of chromosome domains in our genetic map for chromosome 2, where we have low recombination in the center and high recombination in the arms. And in here we observe the proportion of the different founder haplotypes for each of these reals. So then we wanted to know, characterize the fitness measurements of our rails. So we looked at growth rate and sex-specific size. In order to do this, Tom Paré and Erin Kagler from our lab did a lot of really good work to phenotype our rails by 2 methods, by measuring growth rate of a population on a scanner over a set of days. So for that assay, we started with 4 founding individuals, 2 males and 2 females, and we counted the number of hours that it takes for the plate to starve. And then they also looked at the size and sex ratio of those worms by using image express plate scanner, and they also used cell profiler to be able to distinguish between male and female and to measure the length. And this is what they found. In here on the x-axis, we have the different rails. Each vertical line is a rail. And in here on the y-axis, we have the number of hours of before starvation. And all of these rails are from a single cross A. And on the right, we have the worm length for those rails according to their sex. And in here, they observe like a higher worm length for the females than on the males. And using this data, Tom was able to do GWAS for growth rate, size, and size, and he was able to find that growth rate, there is a peak on the first chromosome, and we're still working on this data. We don't really understand much of this, but I'll keep you updated, and we can go over it at a different conference. So what we know from our early days, so what we know now is that our real segregate unique mosaic founder haplotype blocks, and our resolved founder haplotype blocks show that each reel underwent 5 recombination events per chromosome during derivation, and that our reels show distinct replicable phenotypes, and that imaging our reels allows us to discover growth rate for the recovery of growth rate and sex-specific size, and for us to do GWAS using those traits. So what we want to do next is work on a recombinant timbrate line process and develop our genetic basis for fitness, but I'm also interested in looking at tracking the haplotype frequencies all throughout our experiment in the construction of our recombinant timbrate lines, going from the founders to the pools to the reels. And then also I want to examine the patterns of residual homozygosity and residual heterozygosity throughout our genome of our set of recombinant timbrate lines. And then I also want to compare the growth rates and haplotype frequency change during the outcrossing phase and the line derivation phase. And with that, I would like to thank our lab and all of the good people that did a lot of really hard work for this. And thank you for listening.
Get started. So, hello. Despite what the sign said originally outside, my name is David Hubert. I am a postdoc in Mark Phillips' lab at Oregon State University. There we go. And I'm here today to talk to you about our work looking at selection, or populations that were selected for early reproduction, and how that's affected the metabolome and potential signals of aging in these experimentally evolved populations. So first, to talk a little bit about the populations that we're using. So experimental evolution was used to make these evolved populations previously that are made to test the theory of aging and certain aspects of it. And so we have a couple different populations here, or selection regimes. We have the A-type, which have been selected for early reproduction, versus the C-types that have kind of a relaxed selection regime. And so within the A types, they have a 10-day generation cycle, so really short for fruit flies. And the C types have a 28-day generation cycle, which is fairly relaxed. And so the theory of aging would suggest that these A types should have some consequences to this. They would, obviously develop fast to be able to do this, but there should be some increased mortality rates, some reduced longevity within this as kind of this cost and trade-off for earlier reproduction versus longevity. And that's what's been found, right? So we see this really, really super fast development that happens. So these things the A types lay their eggs before the C types are even fully adults. And the consequences of this is that they are dying a lot younger, they have higher mortality rates much earlier, and then they also have things like decreased lifetime fecundity and stress resistance throughout their lives, right? So we have a lot of costs associated with this that match kind of what our predictions would be. So we've looked at this in a lot of different ways. We have these kind of base phenotypes that we know a lot about that you can easily measure. And we've done some work looking at genomic work and transcriptomic work to try and see kind of the underlying reasons for these trade-offs and what's going on. However, a lot of this work has been really hard to make kind of mechanistic insights into like really what's going on. We know that there are differences, but how those differences actually act on these phenotypes is what's been kind of elusive so far. So we wanted to find a kind of a molecular phenotype that was much closer to these observed phenotypes and see if that kind of helps add some mechanistic insight. So we focused in on the metabolome and looked at how that changes over time within the groups and how it compares between the groups. So for this figure, this is mortality rates for the two different populations. So the A types, those early reproducing ones, are red, and the blue is the kind of control groups. And we see that there is just a much higher mortality rates much earlier in the A type. So this is something we've already known. This is pretty expected there. So to do the metabolomic study, we wanted to do a targeted metabolomic study looking at, again, those populations over time as they age and compare between the two of them. So we had 4 time points for each of them with those yellow bars representing these overlapping time points where we have the same time for each. Again, time, sorry, I didn't say this, is age from egg in days for each of the populations. So this is across the lifespan of one lifespan, basically, for them. And then for the A types that develop much faster, we have a time point that gives that really early snapshot, but the Cs aren't actually adults by that point, and then the much later time point for Cs where the As are no longer alive. So we kind of have these different spots we're comparing. So we did targeted metabolomics for this, and in the end, we ended up with 202 metabolites that were included in the study that we were able to look at. So the first thing we did was, obviously, we ran a PCA to kind of see the shape of the data for the metabolites and see if there was any real signal within the metabolome for the different selection regimes and for aging. So we're actually looking at 2 different variables there. And so we see for those common time points where we got samples for both populations at the same time, we do see there's a separation. So we see a good selection effect happening where they are different. The metabolomes do look different during these common ages. But then for day 9, where we only had the C types, where they're that super fast development and they're very young, their metabolome looks very different than everybody else's. So something is going on there that seems striking already. And then for that group, the oldest in the C populations, we see that their metabolome looks much more like the rest of the A types and different than the rest of the C types. So there's a few different things going on here where we're seeing some selection and aging signals already. So that's good. We're starting to see what we're looking for. However, it doesn't really tell us much about how that actually reflects on the aging. So to look at that, we constructed these metabolomic clocks using elastic net regression models, with the idea being that we could build these models that would use metabolomic data to predict the ages, effectively like a biological age versus a chronological age of these different populations to see if they look different at the metabolome when you could predict different ages. So these models predict age we had 2 approaches. One was a within regime approach where you would take, say, for example, the C-type metabolomes, you would train a model on that to predict other C-type ages, and then we would then do a cross regime where we'd use the C-types to predict the A-type ages. And we did them both ways kind of for all the comparisons. Once we predicted the ages, we then ran fit linear mixed effect models to those outcomes to kind of characterize what was going on, see if we saw signals of aging or just signals of selection and all those things. To compare the 2 different model outcomes. So this one was the results for the model built off the C types, so it shows both the within regime and cross regime predictions. And so initially, we found that there definitely was a signal of aging within the metabolome, which is great. This is something that, if that didn't work, none of this worked. But it was pretty strong, so the Cs were able to accurately predict the age of other Cs. We got on the x-axis, we have the actual age in days, and the y is the predicted age from the model, so that's kind of the outcome. So it was a good fit there for that. When we look at how the Cs effectively see the A types, they generally see them as older, and it's actually really interesting here. They see them as about, on average, 29 days older at any given age, and 29 days is about the time at which the C types start to show strong senescence, right? The mortality rates start to increase there. So basically, they are at any given time seeing the A's as already getting old or already aged. But also interestingly, when we look at kind of the rate of aging, they don't see them as aging any faster. They just see them as statically older than them. So that was a fairly interesting result there. When we look at the models trained on the A-type data, we see a little bit different patterns. So they were still able to predict their own age pretty well, which is good. So there was still a strong signal of aging generally within the metabolome. When we look at how they view the age of the C-types, it gets a little complicated, so, and we'll get to why in just a second, but generally when you look where we do actually have age samples for the C-types, the A's tend to see them as younger, like across the board when the samples are actually there. However, it's the rate of aging that they see that actually kind of messes up the exact values of the model. And so as we see the A-types see the C-types as aging more slowly. And when you get to the point where you don't actually have samples for the C's anymore, they're too young, it kind of messes it up a little bit. But we really get that signal of a different rate of aging here, and then also a difference in perceived age. So this is good. We saw signals of aging and differences in age caused by the selection and just age in general, but this doesn't tell us much about the actual specific metabolites that are shaping this kind of accelerated aging phenotype that we observe. So to look at that, we did a linear mixed effect model to kind of disentangle the elements of age and selection and the interaction between the two and see which metabolites are most affected by those different variables. With really the idea being we know that age should show up because we are looking at populations as they age over time. So we want to really pull out what is selection doing in this process? How are we getting a difference in aging? And so this is just giving you the number of metabolites that were significant for the different terms, so either interaction, age, or selection. And as we predicted, 174 metabolites were found significant within this model out of the 202 that we were using. So we got most of them, which is good and also expected because it was a targeted approach, so we should have gotten most of them in it. Of those, only 15 were only significant for age, meaning that age should be there throughout, but only 15 of those didn't involve selection in some way in their significance, and so that means the other 159 did have selection play a role as to why those were significantly different. So that's also awesome. So that kind of gives us which ones are important in this process. It doesn't really tell us much about the trajectory or how they're different and how things are changing over time. So to look at that, we did some comparisons where we're looking at kind of relative abundance levels within selection regime and how those change over time, so kind of between those time classes. So this is a heat map. Just think the red is higher change, blue is lower, and I'm going to explain it right now. So if we zoom in on just that one cell, so this is glucose, that's the metabolite we're looking at, and this is the abundance between on day 9 and day 21. It's a normalized relative abundance, and we see that it increases dramatically between day 9 and day 21. That gives you that large positive log 2-fold change. So that's kind of how this whole table is breaking down. And we made some predictions about kind of the trajectories we should see in these aging populations. And there's two good examples here. There's a ton of stuff we looked at. These are just some highlighted ones. But if we look at the top section here, what we're really seeing, we kind of call this an example of an accelerated phenotype, right? It's an accelerated aging, where the same basic pattern is occurring in the A and C types, but it's happening sooner in the A types than it does in the C types. And so we get this kind of accelerated shift. The other side here is that we just have different. It's not accelerated, it doesn't match in any way, shape, or form. It's just something different is happening with these metabolites between the two different groups over time. And to kind of pick one that we find interesting, we're going to focus in on the carbohydrates here. And if you see the part where at that difference between day 9 and day 21, we see a huge change in the same kind of way within the A types that's not characterized in the C types. And so we want to try to think about what that might mean in the broader picture, how that might connect to these bigger phenotypes that we've seen. So to go back to a couple things we said already, remember the day 9s and the A's that were really, really different, right? They have a very different metabolome than everybody else. And if we look back at the experimental design, right, we have this day 9 is going to be the 24 hours where they're adults before they get the chance to lay eggs. They don't get to lay eggs after that window. So we have this very small window that reproduction is involved before the selection comes into play. If we look at some previous work that's been done, while I said their overall lifetime fecundity is much lower, their acute fecundity in that window where they get to reproduce is 2 to 3 times that in the A's that it is the C types for their reproductive window. So we see a lot of eggs produced in a very small period of time that coincides with the other things that we're seeing here. And then other work also has found that there is some pretty significant metabolic remodeling involving glycolysis when it comes to egg production in female fruit flies. So they change a lot to really ramp up glycolysis to produce these eggs. So overall, when we put that all together, what we really think we're seeing as a small piece of this is that this optimization for early reproduction from the selection regime is causing a change in energy substrate use to prefer or to use that to produce lots of those eggs really, really quickly, right? So we think that's the big trade-off there. And with some other stuff that I wasn't able to show because of time, we also see some changes to amino acid use that reflect aging that we see typically across the board. And so we really think that there's this trade-off where they're shifting to produce eggs and that there's the cost where they have this breakdown, metabolic breakdown that causes these kind of other phenotypes that we see with the accelerated aging. So, general conclusions, right? So we did see a metabolomic signal of aging caused by selection for early reproduction, right? Those signals include both the accelerated aging and kind of differently aging phenotypes. And kind of more specifically, we're hypothesizing that selection for early reproduction is resulting in this metabolic remodeling for egg production, and that is going to lead to glycolytic dysregulation later down the road. And we have some stable isotope tracing things planned to kind of figure out the fate of glucose in this process to actually see if that's what's happening for sure. I'd like to acknowledge our collaborators in this, our funding sources. This actually is in review right now. We did a lot more than what I was able to present, but this is a QR code for the preprint. If you want details, I'm also happy to answer questions and talk about it. And if you need to reach me at all, this is my contact, and that's a QR to my website. So with that, questions? So we only looked at females for this. So yeah, I guess I didn't say that part, but yeah, so we wouldn't have found that.
Just another question is that, did you find any changes during their reproductive schedule, like in the A type and the C type? If you can compare at all that, the 9-day for the A type and the 28 days for the C type. So did you compare the metabolic pattern between these two days?
Like just at their reproductive window? I did a little bit. It gets difficult with the age separation to make super specific comparisons, but a lot of it was just very different. We see that the A types at that day 9 seem to be very optimized, very different metabolically than everybody else. Yeah.
Thank you.
Yeah.
Thanks for a lovely talk.
Thank you.
So you found this glycolysis dysregulation, right? Did you find any signatures downstream with any sort of Krebs cycle pathway or anything like that?
So because within the targeted thing, we only get certain pieces, so there are some pieces missing in what we're able to look at. But things like NADH and some other stuff and pyruvate kind of stay stable throughout, and we don't see the same drop that we see. So yeah, it's interesting. Yep. Anyway, thank you.
Okay. Hello everyone, I'm Claudia Pruvôt, PhD student at the Institute of Functional Genomics in Lyon, France, and I'm going to present a part of my PhD project, which is on the evolutionary mechanisms underlying leg growth variation in Microvillia longipes. So as you know, a phenotype is the result of a genotype of an individual the environment in which he is living, and the interaction between the two of them. And at a populational level, a same trait can have different phenotypic values. So we are talking about phenotypic variation, and here is a simple example where the trait is the color of a flower of snapdragons, and you can see that it goes to yellow from magenta. And some traits are really markable by how diverse they are, and sexually selected traits are among them. They are described as being more phenotypically variable than any other traits, and sexual selection can act on the phenotype of an individual through different mechanisms, such as mate choice in peacocks or intrasexual competition in red deers. Sexual selection is directional. It can lead to the exaggeration of a trait, as you can see here, and it can lead to the fixation of a phenotype, which is paradoxal with the fact that they are extremely phenotypically variable. And this is what we are interested in our team, is to understand the different mechanisms underlying this phenotypic variation associated with sexually selected traits. And for that, we are working on a water strider species called Microvillia longipes. And as you can see, there is sexual dimorphism in this species. The size of a third pair of legs in males is exaggerated compared to his body size, whereas in females, the size of a third pair of legs is proportionate to his body size. And in wild population, this phenotype is extremely diverse. You still have his exaggeration of a trait, but there is really huge male and small one, and it follows a Gaussian distribution. This trait is under sexual selection, and I will explain you how. So on the first page, here you can see a female. She seems to put into the object, and there he is male, mate. And in the natural habitat, females are always looking for this kind of object that they use as egg-laying sites, and males are waiting by them to mate with females. And on this second footage, a male is guarding an object, another male is coming, and they use their third pair of legs to kick each other. So they fight over these egg-laying sites. And in the team, they showed that males with the longest pair of legs is most likely to win a fight, and so to have access to more females and fertilize most of the eggs. So they wanted to know if there was genetics or influence of the environment. So here on this graph, you can see the distribution of the size of a third pair of legs in 3 different populations. So the red curves is the white population, and the 2 gray ones are isogenic lines created by the team. And you can see that in doing so, they succeeded in splitting and shifting the 2 distribution, and so each isogenic lines have a specific phenotypic distribution, so there is a link between the genetics and phenotypic variation. And then they did the opposite. They took one of these isogenic lines and put it in 2 different environments. So here, a nutritional environment during non-fold development, and you can see that there is also two specific phenotypic distribution. So there is phenotypic plasticity in this trait. And then we want to dig a little bit further in the mechanisms underlying this phenotypic variation. And so all that is linked to phenotypic plasticity is done by other members of a team, while I'm focusing on the genetic variation that can explain leg growth variation, and also the role of different ecological factors in driving this extreme phenotypic variation. But for today's presentation, I will only present the first point. So as I said, I'm focusing on the genetic contribution to this phenotype. And we want to establish the heritability of the trait, to know how much genetics contributes to this trait, but also to see how changes in allelic frequencies will impact this phenotypic variation. And for that, we are doing an evolve and resequence experiment. And this is this last point I will present. So with this approach, to see how which alleles are involved in this phenotypic variation. But for that, at the beginning, we have a base population, so the G0. in this population, we have a certain distribution of the size of third pair of legs and also genetic variability. We will apply selection pressure in favor of mares with long legs over several generations. And at several time points, we will measure the phenotypic and the genetic response to this selection. And at the end, we are expecting to have a shift towards mares with long legs and have alleles that correlate to this phenotype. So for creating this base population, we decided to use a wild population, so to have a greater phenotypic and genetic variability. And it is a tropical species. So last year we went to French Guiana to collect some animals. So they lived in puddles formed by rain, so very temporary habitats. And you can see on the first pictures there is males and also females, and they live together all year long. And on the map here is the different sampling areas where we collected animals. And we took these animals back to the lab. So the green distribution is the distribution of the wild population that we collected last year. And we let this population mate for one generation. And we took this as our base population, or G0. And it's the orange distribution, so you can see the same distribution, so there is no real impact on the lab on the trait. And then we took this base population, we split it in two conditions, so selection conditions, where we want to select for mares with long legs, and the control conditions. And as I said earlier, there is phenotypic plasticity on this trait during non-full development due to nutrition. And we don't want that because we want to select for a specific phenotype and specifically to have a link with the genotype, so we want to get rid of phenotypic plasticity. So we decided to do a nutritional treatment and expecting to have a strong link with between phenotype and genotype. And here are some preliminary results of G0 and G1. So the base population, you can see that the distribution between the two conditions are the same, so there was no bias when we created the two conditions. And you can see that the G1 is different from G0 for both conditions, and it's due to the nutritional treatment. So control are a bit bigger because they were under rich diets, and selection were the poor diet. And for the following generation, we're expecting that the control will be the same as the first generation, because the nutritional treatment will be the same. And then when they reach adulthood, we stop the nutritional treatment. They are under the same diet for both conditions, and this is where we will apply the selection pressure in favor of mares with long legs. And to do that, we need to increase the competition between mares, and we will play with the size and the numbers of floaters, which are the resources that they are fighting for. And so the floaters are like the size of one individual, so that one individual can defend it, and they are in really low numbers, so the resource is rarer, and it will increase fight. And in the control condition, the floaters are really huge, so they can not, one individual cannot defend it, and so it's supposed to be a relaxed selection. And here also some preliminary results. At G11, we decided to take, I forgot to say that then here we took the eggs that are laid on floaters and we put it in new buckets and repeat the same thing generation after generations. And so at generation 11, we took eggs that were in excess, and we put them for the two conditions in the same nutritional treatment, so that we only have the effect of selection and that we can truly compare them at the phenotypic level. And you can see that there is a difference between the control and the selection. Males in the selection conditions seems to have legs bigger than in the control conditions. And it's not here, but the base population is similar to the control control condition. And it's really preliminary result because it's only one replicate per conditions, and I have 4 replicates. So, but it seems to work. So, yeah. So here I only have phenotypic results because we only received the sequences last week. So no sequences. Data yet. And so for the future direction of this project, we want to automatize the measurement for the phenotypic data because it's really long. This is why it's only a preliminary result, because I have thousands of individuals per generation, so it's quite long. And yes, I need to do the analysis for the sequencing data, and then when we will have find the alleles, the genes that are involved in phenotypic, in this phenotypic variation, we want to do RNA-seq to see if the genes that are expressed during development are the same that are targeted by sexual selection. I would like to thank you for your listening, and also thanks my lab's members, and happy to take any question.
You told us how the males have the extremely exaggerated long legs might gain mating success. But you've got this genetic variation, and I just wonder when you see that, it sometimes suggests there might be an alternative reproductive tactic. Do you consider that there might be a way for short-legged males to gain mating success?
Yeah. We see that in our buckets and also in the whites that small males succeed also in reproduction, but sneaking behavior and kind of like that. But yeah, it's not on these egg-laying sites, so it's not the one that will develop. And we don't know much about the dynamics of this population in the natural habitats. But we suppose that small males will mate with females, and these females will lay eggs in the mud. So since it's temporary habitats, not all of them will survive. And yeah, but we think that